Welcome to Science Made Simple, where we discuss scientific topics and break them down so that anyone, regardless of your level of scientific knowledge, will leave with a better understanding of science than before they clicked on this video. Today, we'll be talking about one of the many unique processes that occurs every second in your body, known as cellular respiration. Just on a side note, this video will also answer questions like why breathing oxygen is so important, and how exactly cell respiration helps us to function at an optimum level. These are the essential questions. Cell respiration has three main steps and an intermediate step. The three phases are known as glycolysis, the Krebs cycle, also known as the Citric Acid Cycle. The third phase is the Electron Transport Chain, also known as the ETC. The intermediate step is known as Pyruvate Oxidation, and it occurs in between the glycolysis and the Krebs cycle. The main purpose of cellular respiration is to generate energy, specifically 32 to 38 Adenosine Triphosphate, also simply known as ATP. ATP is a key energy molecule that our body generates to use for its own needs, such as breaking down food, so food breakdown, moving your muscles, so muscle contraction, and for CAMP conversion to provide protein kinase activity via signal transduction, also simply known as cell communication. There are two main types of cell respiration. One is aerobic, the other one is anaerobic. As you probably already know, aerobic refers to the presence or need of oxygen, whereas anaerobic doesn't need oxygen. So you can already tell that we'll need oxygen in some capacity for this process to occur. It is how we'll answer why oxygen is so necessary for the body and why exactly animals, humans, and other organisms use oxygen to breathe, and what the purpose of cell respiration is in the first place. We'll answer all these questions in this video. And although these two processes, they end up making energy, they end up making ATP, anaerobic respiration has a slightly different mechanism to make energy than aerobic respiration makes energy. So we'll discuss anaerobic respiration in a later video. Today's video is about aerobic respiration. And just before we get into the details, the key equation you must know to fully grasp and understand cellular respiration is 6O2 plus C6H12O6 yields 6CO2 plus 6H2O plus ATP. As you guys know, this equation refers to the cell respiration reactants and products, which are 6 molecules of oxygen, 1 molecule of glucose, which leads to 6 molecules of carbon dioxide, 6 molecules of water, about 32 to 38 ATP, depending on the literature or the textbook that you're reading from, or the teacher that you're getting your information from. To make it simple for this video, we'll just go with the maximum amount of energy and say that cellular respiration generates about 32 ATP. Again, this might be different for you, depending on the source that you're getting your information from. As we mentioned earlier, the first phase of cellular respiration is glycolysis. Glyco refers to glucose, and lysis refers to breakdown. So glycolysis is just a fancy scientific way of saying glucose breakdown. So it occurs in the cytoplasm, or the cytosol of the cell, which is just the liquid portion of the cell without the organelles. So we start glycolysis with the molecule of glucose, C6H12O6, and it breaks up into two pyruvate molecules. So pyruvate. And these two are further used down the cell respiration reaction. Pyruvate is also known as pyruvic acid. An easy way to understand the relationship between glucose and pyruvate is that glucose is a 6-carbon molecule and pyruvate is a 3-carbon molecule. And then this is also 3-carbon molecule. So two 3-carbon molecules of pyruvate make up one 6-carbon molecule, such as glucose. We start with 2 ATP to start glycolysis, which further yields 4 ATP. So while 4 ATP is generated, 2 ATP is initially used, so the net is production of ATP after glycolysis is 2. So 2 ATP is generated overall. In this process, a molecule known as NAD positive or plus is converted into NADH. NAD is an electron carrier, so we'll write NAD is an electron carrier. In glycolysis, 2 NAD positive or plus is converted into 2 NADH. This process is known as reduction. To put it simply, reduction is a concept used in general chemistry to show a gain of electrons by another molecule. The opposite of this process is known as oxidation. Oxidation is just a fancy term that means a molecule loses an electron to like another molecule or substance. This is the reason that NAD makes for a great electron acceptor. It can undergo reduction. NAD positive or plus reacts with a hydrogen ion and an electron to convert into NADH. This sums up the aerobic portion of glycolysis. So glucose is broken down into 2 pyruvate molecules, 2 pyruvates. Then you have the production of 2 NADH, and you do have 4 ATP generated, but you have 2 net ATP produced. So this is the reactant, and these are the products of glycolysis. Glycolysis. You might remember that I mentioned earlier about an intermediate step in cellular respiration. This step is known as pyruvate oxidation. Pyruvate oxidation occurs in the mitochondrial matrix, the inner portion of the mitochondria. Remember, this is not a really great representation, but the mitochondria has an outer membrane and it has an inner membrane. And this process begins with like pyruvate, as we said before. So, and as the step shows, pyruvate is getting oxidized. So pyruvate, which is again a 3-carbon molecule, it becomes catalyzed. It catalyzes into an acetyl group, which is a 2-carbon molecule. The enzyme that helps catalyze this reaction is called, so let's write over here, enzyme. And as we know, enzymes catalyze many reactions. So they're very specific. This specific enzyme is called pyruvate dehydrogenase. Pyruvate dehydrogenase. Since 2 pyruvates are generated per glucose. This oxidation step happens twice. But you might be wondering, what happens to this acetyl group? How does it go from a 3-carbon to a 2-carbon molecule? What happens to the other carbon? As pyruvate is oxidized, one of the carbons is expelled as CO2, also known as carbon dioxide. And again, as the step happens twice, it's going to expel 2 CO2, right? It's going to happen 2 times. This also involves an NAD plus becoming reduced and converted into NADH. Overall, in pyruvate oxidation, we have a pyruvate, which is the reactant, and it's converted into 2 CO2, 2 NADH, and a coenzyme group is actually attached to the acetyl group. So let's say right here, you have a coenzyme group. So now you have a production of acetyl-CoA, acetyl-CoA. And now that we have acetyl-CoA, we can finally start the Krebs cycle. So, as acetyl-CoA is produced, we then begin the Krebs cycle. And just like pyruvate oxidation, this occurs in the mitochondrial matrix, which is the inner portion of the mitochondria. Then you have acetyl-CoA, acetyl coenzyme A, which is our 2-carbon molecule. It attaches to oxaloacetate, also known as oxaloacetic acid, which is our 4-carbon molecule, and it forms citrate, which is a 6-carbon molecule. Citrate then undergoes a series of biochemical reactions, and it undergoes oxidation. And through oxidation, 2 carbons are cleaved off of the citrate molecule. And again, oxidation just means a molecule loses an electron and is transferred to another molecule. In this case, these electrons that leave the carbons during oxidation attach to NAD+. And then, once the electrons attach to NAD+, it is reduced to NADH, which, remember, they're electron carriers. Similarly, there is another type of electron carrier that we now find out about. It's called FAD. And it also attaches to a few electrons, and it's reduced to FADH2. Through oxidation, these 2 carbons that lead the citrate end up forming 2 more molecules of CO2. Also, ADP, adenosine diphosphate, is then phosphorylated. It's phosphorylated into ATP. So phosphorylation just means the addition of a phosphate molecule. You have a diphosphate, which means 2 phosphates, and then you phosphorylate it by adding a phosphate, and now you have a triphosphate, which is 3 phosphates. As we mentioned before, NADH is an electron carrier, which will be more useful in the electron transport chain that we will get to shortly. FADH2 is also an example of an electron carrier that accepts electrons when it is in the form of FAD. There's one key difference between these 2 molecules, which we'll get to as we discuss the role in the ETC. That difference is that each NADH is responsible for the production of 3 ATP. And each FADH2 is responsible for the production of 2 ATP. And we'll get into why that is. So just to recap, the Krebs cycle produces about 4 CO2, 2 per pyruvate, produces about 6 NADH, 3 per pyruvate, 2 FADH2, 1 per pyruvate, and 2 ATP, also 1 per pyruvate. Pyruvate oxidation, on the other hand, produces 2 CO2, 1 per pyruvate, and 2 ATP, 1 per pyruvate. So remember that glycolysis as well. Glycolysis produces 2 NADH and 2 ATP, both of them 1 per pyruvate. It is important to know how, the distinguishment between each pyruvate versus the total amount. Students sometimes often get confused on where these numbers come from. You have 2 pyruvates from glycolysis, therefore you have to double the cycles. And so in total, you have 10 NADH, 2 FADH, and 4 ATP heading into the electron transport chain. The electron transport chain. The remaining 6 CO2, remember the equation, 6O2 plus C6H12O6 yields 6 CO2 plus 6 H2O plus ATP. The 4 CO2 from Krebs cycle and 2 CO2 from pyruvate oxidation both combine to 6 CO2. So all the carbon dioxide in the whole respiration process occurs from Krebs cycle and pyruvate oxidation. All 6 CO2, and they'll all be released as waste product. And the remaining 10 NADH, 2 FADH, and 4 ATP are used in the ETC. And now we enter the final stage of cell respiration, known as the electron transport chain. This is the phase of cell respiration that generates the most amount of ATP. This process also occurs in the mitochondria. But instead of the matrix, it occurs in the cristae, which are a bunch of little folds in the inner membrane of the mitochondria. To help understand this, let's just draw it visually. Here is the outer membrane of the mitochondria. And this is the inner membrane of the mitochondria. It has a bunch of little folds so we can demonstrate the cristae on the outside. So the inner area over here, that we just colored in blue, is the matrix. The matrix is where the Krebs cycle or the citric acid cycle and pyruvate oxidation happens. The cristae is where we just said the electron transport chain occurs. So we start off with 10 NADH, and I'll abbreviate here. We get 2 from glycolysis, 2 from pyruvate oxidation, and we get 6 from the Krebs cycle or the citric acid cycle. We also have 2 FADH2, and both of them we get from Krebs. As we said before, each NADH produces 3 ATP. Each FADH2 produces 2 ATP. The reason why that this is because the electrons in the NADH have more energy than they do in the FADH2. And as we said before, both of these molecules, NADH and FADH2, are both electron carriers. They can both be oxidized and can revert back into NAD plus and FAD. Both of these molecules can go back into glycolysis and the Krebs cycle to again be reduced again into NADH and FADH2. So here we have a picture of a mitochondria. Here's the outer membrane, the inner membrane, the intermembranous space, and the protein channels that allow the hydrogen cations to move through. And here are the fold, or the cristae, as we tried to show over here as well. So when NADH and FADH2 get oxidized, as in they lose electrons, these electrons then bind to specific protein molecules within the inner membrane, such as coenzyme Q or cytochrome C, for a very short amount of time before these electrons then attach to hydrogen and incoming oxygen to form water. As the electron bounces around from NADH to coenzyme Q or cytochrome C, it loses energy, and it is this energy that powers protein channels in the cristae to pump out hydrogen cations from the matrix into the intermembranous space. Again, as the electrons from NADH and FADH2 move around to different molecules, it is losing energy, which then powers the hydrogen ions out into the intermembranous space from the matrix. Eventually, hydrogen ions just hang out in the outer compartment of the mitochondria and then want to re-enter the matrix. They want to re-enter the matrix because they are in a concentration gradient. Hydrogen ions move from where they are large in number outside to where they are small in number now inside. But they can only do so with the help of a special protein channel known as ATP synthase. ATP synthase. But ATP synthase is not like other protein channels. It has a special structure. There's a tube-like structure that is centered within the synthase molecule that moves around as hydrogen ions flow through. So as hydrogen ions re-enter the matrix, this tube-like structure moves around like a windmill. This tube has all sorts of positive, negative, positive charges, and that they move around as the protein is moving, and it attracts different molecules. These charges attract different molecules as protein is moving. One group of these molecules are ADP, adenosine diphosphate, and a phosphate group. As the hydrogen ions move through the ATP synthase, as we said, different charged molecules move around and they attach to each other. So as the hydrogen ions move through the ATP synthase, ADP and the phosphate group attach to each other to form what we know as ATP. Literally, the molecule name is ATP synthase. It's synthesizing ATP. So we have about 10 NADH from the first 3 steps of cell respiration to FADH2 from Krebs cycle. We also produced 4 ATP. So, since 3 ATP is generated from 1 NADH, you have a total of 30 ATP because 10 times 3 is 30 ATP. 30. Then we have 4 ATP generated from FADH2 because if one FADH2 generates 2 ATP, then 2 FADH2 is going to generate 2 times 2, which is 4 ATP. And then, since we already have 4 ATP from glycolysis in the Krebs cycle, we're going to generate 4 more ATP. And so this generates about a grand total of 38 ATP. At the beginning of the video, I asked the question about why we breathe oxygen and why it is so important to breathe oxygen. Remember when we talked about the electron's energy powering the protein channel to move the hydrogen ions to the inner membrane space? Well, what happens to the electrons once they lose the energy? Well, they become what we call free radicals. And free radicals can become really dangerous to the body as it can damage blood vessels and do other things to hurt our body. This is the sole reason why we breathe oxygen. It is to capture that free electron, the free radical electron, and join it with the hydrogen. So this oxygen that we get from the 6O2 of our equation, it is to capture this electron and join it with hydrogen to form water. ATP production also helps maintain our energy levels so that we can function properly, such as muscle contractions, cell communication, etc. And so let's draw our equation again. Let's write our equation again. You have 6O2 plus C6H12 12O6 yields 6CO2, which we already discussed, that was our waste product, plus 6H2O plus ATP. So we already made our ATP. Now we already have water because of this oxygen. CO2 is already left, and glycolysis already happened for the glucose. We have already talked about each single component. And that is what cell respiration is. If you thought this video was helpful and can help someone else, leave a like down below and share this video to help make science more simple. Subscribe for more, and we'll see you in the next one.